Okay guys, so I'm going to go through this pretty quickly, and if you have questions you need to have come in, well, today, after school on Monday, or come in Tuesday before school. Alright, so a calorie is a unit of, well, it's a unit of energy. There's not much I can explain about that. The calorie used on food labels is equal to, remember, the big calorie equals 1,000 little calories. Okay? Cells use the energy stored in chemical bonds of foods to produce compounds such as, well, ATP is what is specifically being formed, because ATP is used to power all cellular processes, so pumps, the flagella and bacteria, everything. Okay? We store energy in glucose, but then when we're actually needing it to use, we make ATP. Number 4, this is just the formula. Remember, the reason why I have the 6's there, it is to balance. So I've got 6 O's here, I've got 6 times 2, which is 12, so I've got 18 O's here, and then 6 times 2, there's 12, and there's 6 here. So I have 18 O's on both sides, I've got 6 carbons on both sides, I've got 12 hydrogens, 12 hydrogens. Okay. You won't need to actually balance, but I just wanted to show you where it came from. All right, the next one. Cellular respiration begins with a pathway called, well, glycolysis, and this takes place in the cytoplasm of the cell. Okay, we'll go over that more in a little bit. Cellular respiration continues in the mitochondria, so you can see it right here. It's happening in the mitochondria, and this involves both the Krebs cycle and then also the electron transport chain. The pathway of cellular respiration that requires oxygen are said to be aerobic, while the pathways that do not require oxygen are anaerobic. Okay? And here are the 3 steps of cellular respiration. You've got glycolysis, the Krebs cycle, and the electron transport chain. All right, so in this, guys, what this is asking you to do is to list your products and then circle the ones that are then used in the next step. So for example, in glycolysis, NADH, ATP, and pyruvic acid are produced. All right. Pyruvic acid is then taken and used in the Krebs cycle. So that's why I drew that arrow. Now, NADH and ATP are not used in the Krebs cycle. They are going to be later used in the electron transport chain. For the Krebs cycle, the products are NADH, FADH2, ATP, and then also CO2. Okay, NADH and FADH2 are what are actually used in the electron transport chain in addition to the NADH here. Okay. But ATP is not. ATP is actually then later used by the cell for energy, so it doesn't go into the electron transport chain. CO2 is just released from the cells. It's a byproduct. We don't need it. That's why we exhale it. For the electron transport chain, your products are ATP and H2O, which are the most obvious. And then since NADH is going in and delivering electrons, NAD plus is going to be made, and FAD is going to be made as well. These guys are important because, and I'm not sure why I drew that arrow there. Oh, I know what I did. NAD plus and FAD are then going to be taken back, and they're the ones that collect electrons from glycolysis in the Krebs cycle. These guys are necessary in order for these guys to be made. So if this stops, guys, you've got a huge issue because you need a way to replenish the NAD plus and the FAD. So if the electron transport chain isn't happening, meaning I don't have oxygen, this is automatically going to stop. And then we have to find a different process for this to keep going. And that is what fermentation is. Getting the NAD plus back, because you're creating NADH, you need NAD plus to do that. All right. Photosynthesis deposits energy, that's true. So the answer for this one is that cellular respiration takes place in nearly all life, right? Plants respire, they have cellular respiration and photosynthesis, but we don't have photosynthesis, obviously. The pyruvic acid produced in glycolysis enters the blank, well, it's the mitochondria, because that's where the Krebs cycle starts. In the matrix, pyruvic acid is converted into acetic acid. That's your 6-carbon molecule. Remember, pyruvic acid is broken down into a 2-carbon molecule called acetyl-CoA. It joins with a 4-carbon molecule that's already in the Krebs cycle to make acetic acid. And guys, if this is way over your head and you're not ready for this explanation, stop this video right now. Go back to the other one where I explain the entire process, because I go through all of cellular respiration. All right, a compound joins with a 2-carbon molecule in the Krebs cycle to make acetyl-CoA. That's correct. Coenzyme A joins with a 2-carbon molecule to make this. Okay, 14, carbon dioxide is the only product of the Krebs cycle that is not reused or used later. That's true. Remember, my explanation here, you've got NADH and FADH2 being used in the electron transport chain, and ATP is going to be used in order to power the cell. So CO2 is the only kind of useless one. It's just a byproduct. Glycolysis provides the pyruvic acid molecules used in fermentation. Okay, that's true because you start with glycolysis and then the pyruvic acid is then going to be broken down into either an alcohol or into an acid. Okay. Fermentation allows glycolysis to continue by providing, it's not NADPH. First of all, that's for photosynthesis. That's totally wrong. Fermentation is taking the NADH, which is produced in glycolysis, and making it into NAD+. Okay, so that was false. This is what matters because then it'll grab electrons from your glucose, from your PGAL, in order to make NADH. Fermentation is an anaerobic process. That's the whole point, because the Krebs cycle and the electron transport chain can't happen, and so I need another way to remove the electrons from NADH. So that's what fermentation does. Fermentation occurs in the cytoplasm of cells. That's where glycolysis happens. That's where fermentation happens. Alcoholic fermentation gives off carbon dioxide. That's true. All right. So here, guys, all you're doing is saying, okay, in the Krebs cycle, the two things that are produced that are then used by the electron transport chain are NADH and FADH2. So that should be review at this point. All right, 21. In eukaryotes, the electron transport chain is composed of a series of electron carriers located in the inner mitochondrial membrane. And I think I do it, I'm trying to find where I made my drawing. I can go up here and show you really quickly. Okay, this is your outer mitochondrial membrane. This is your inner mitochondrial membrane. This is your matrix. And then this is your like between two membranes, intermembrane space. Okay, so that's your intermembrane space. Inner mitochondrial membrane, outer mitochondrial membrane. I'll keep going because I'm running out of time. All right, blank serves as the final electron acceptor. That's oxygen. Okay, and I reviewed that in your in the previous video, so go back if you're unsure about that. This is what grabs the electrons at the end of the electron transport chain. Blank and blank pass high energy electrons to the electron transport chain, NADH and FADH2. These are your electron carriers. The transfer of high energy electrons down the electron transport chain causes blank to be transported, H+, right, because they're pumps. So the electrons are powering those pumps. ATP synthase produce the force needed to add one phosphate to each ADP. And for this one, you get 36 ATPs that are produced. All right, so here's how we break down the ATP production. In glycolysis, for every glucose molecule, 2 ATPs are made. In the Krebs cycle, 2 are made as well. And then in the electron transport chain with the ATP synthase, it's 32. Okay. What are 3 main sources of ATP available in human muscles? You've got the ATP that's already present. Okay, and then once you start running and use that up, then glycolysis starts up because it's so fast. But then that can only last about 90 seconds because then you have a lot of lactic acid built up. And then the last step is cellular respiration. So in a race, I'm going to be using first glycolysis if I'm sprinting, but it only lasts about 90 seconds. Okay? And if I'm sprinting, guys, what happens, the reason why I can only last for 90 seconds, in this lactic acid fermentation, a lot of lactic acid builds up in your muscles. And so the only way to get rid of lactic acid is with oxygen. So that's why you're huffing and puffing after a race, because you're needing the oxygen in order to break down the lactic acid. A runner needs more energy for a longer race. How does the body generate that? Well, cellular respiration. And that's why you can't be sprinting for a very long period of time. You need to do cellular respiration, which is slower than fermentation. Fermentation / glycolysis. All right, so fermentation, guys, is much faster, but you get fewer ATPs. Cellular respiration is slower, but you get more ATP. So it's a balance, and we only do this for the first 90 seconds, and then we go to just regular cellular respiration. All right, how do organisms get the energy they need? They have to break down food slowly, guys. They can't suddenly release it all, or else it would be lost as heat. So we slowly break it down. We have 3 steps in cellular respiration. Okay, 3 steps to cellular respiration include glycolysis, Krebs cycle, and the electron transport chain. What are the products of glycolysis? Well, it's all of them. You have pyruvic acid, you have ATP that's produced, only 2 though, and then you have NADH because it's removing electrons. In order for glycolysis to begin, 2 ATP are provided, which then produce 4, so the net gain is 2. What are the benefits of glycolysis? It can be performed under anaerobic conditions, which is important if you don't have any oxygen. Okay? It can make ATP really quickly. It does not produce a large quantity of ATP, so the answer is D. What product of glycolysis is then used in the Krebs cycle? Okay, it's pyruvic acid. So you start with pyruvic acid, and then you break it down into acetyl-CoA, then it becomes acetic acid. Okay, review that other video if you need help with that. Identify the progression of molecules in glycolysis. You first start out with glucose, then it goes to pGal, and then pyruvic acid. In the Krebs cycle, you're going from pyruvic acid, which is given to you by glycolysis, then acetyl-CoA, and then acetic acid. So go back over that video, guys, if you need help. Although only 2 ATPs are produced, the Krebs cycle is most important because it produces blank. Well, it's producing NADH and FADH2, and these give your electron transport chain the electrons that it needs, because when they deliver it, they then become NAD plus and FAD. Okay, so the answer is D. The final output of the Krebs cycle includes all of the following except what? Well, NADPH. Yeah, NADH would be produced, but not NADPH. Okay, that's just for photosynthesis. 44. What is this correct sequence? And you go from glycolysis, then Krebs cycle, then the electron transport chain. That should be a pretty easy one at this point. Which of these is a product of cellular respiration? Water, right? Because oxygen is grabbing the electrons off of the electron transport chain. It's combining it with hydrogen to make water. Which process does not release energy from glucose? Photosynthesis. Photosynthesis is putting the energy into glucose to make glucose and have it be a high energy sugar. So that doesn't make sense. Unlike photosynthesis, cellular respiration occurs in all eukaryotic cells. It occurs in prokaryotes. So C is wrong because it's not only them. We do it too. So D would be the answer. Which of the following is not a product of glycolysis? Glucose. You start with glucose. Right? These are made in glycolysis. This is your reactant. This is what you start with. So that makes no sense. The Krebs cycle does not occur if oxygen is not present, because if oxygen is not present, you don't have a final electron acceptor, and therefore the electron transport chain won't work, and therefore you won't have any NAD plus or FAD to grab the electrons from the Krebs cycle. So the Krebs cycle will stop. In eukaryotes, electron transport chain occurs in A, the inner mitochondrial membrane. Okay, and just look back at that diagram, it explains that. Cellular respiration uses one molecule to produce 36 ATPs. The air bubbles in bread are due to your alcoholic fermentation, which makes alcohol and CO2. Lactic acid fermentation, you're just getting lactic acid and then your NAD plus back. The conversion of pyruvic acid into lactic acid requires blank. Well, it requires NADH, and that's the whole point. It needs NADH, it steals 2 electrons from it, so this becomes NAD+, which then is recycled back into glycolysis. This NAD+ is needed for glycolysis, so we have to get rid of NADH. So that's what lactic acid fermentation uses. Which is not a product of fermentation, oxygen. It's just not. CO2 is produced, ethanol is produced. Oxygen definitely is not. What role does oxygen play in aerobic respiration? It is your final electron acceptor, and it makes water. During aerobic respiration, FADH2 is produced in the Krebs cycle. Your 2 electron carriers are NADH and FADH2. NADH is produced during blank. It's produced during your glycolysis and the Krebs cycle. The reason why it's not produced in the electron transport chain, it's used up in the electron transport chain. You can see NADH here delivers 2 electrons to the electron transport chain, and therefore it's NAD+. Okay, everything else produces NADH. During what stage is the most ATP synthesized? Electron transport chain. What substance is regenerated by fermentation? NAD+. It's the whole point of fermentation, to get NAD+ back from NADH. During chemiosmosis, you're moving from your H+ from the inner membrane space into the matrix back, because you have a high concentration of H+ in the inner membrane space and a low concentration in the matrix. So that's what chemiosmosis is, going from high concentration to low, both concentration and a plus charge in the inner membrane space and a negative in the matrix. Last one, this one, cellular respiration occurs in all organisms. Okay, so D, all of the above. That's it, guys. Rewatch the other video.